Thank you so much.
Yep.
Now, our next speaker is Professor Paul Murphy, Department of Chemistry, National University of Ireland, Galway, Ireland. Professor Murphy's research interests are in the design, synthesis, and evaluation of bioactive molecules, including natural products. A major current interest is in the design and synthesis of glycomimetics to develop the inhibitors of carbohydrate binding proteins such as galactans, macrophage, galactose, C-type, lectin, sigla, and carbohydrate binding proteins involved in infection. Synthesis and evaluation of bioactive. All of these targets are relevant to development for new therapeutics, vaccines, or diagnostics for disease. This is his area of work or interest. Could I request Professor Paul Murphy to please start his lecture? Thank you so much.
Okay. Okay, Ajahn, how'd you get this go? Okay. So can you see my screen?
Yes, yes, we do.
Can you see my slides?
Yes, yes, it is there. Yeah, it is there.
Yeah, I'm showing my screen, but I can't, I'm not sure what you're seeing.
This is the first slide on the synthesis of immunosugars and potential as antiviral agents, which we see.
Okay, yeah. I'm looking at it on YouTube, but oh yes, I see now. Okay. Okay, thanks very much. Thanks very much, Professor Carey and the organizers for the opportunity to speak at this. I'm going to talk about iminosugars. I should just mention that as well as being in the School of Chemistry, I'm also an investigator in the SSPC, which is the SFI Research Center for Pharmaceuticals, and CURAM, which is the SFI Research Center for Medical Devices. So just to outline the structure of the talk, I'm going to speak, to just give a brief overview of our research, and I'm then going to talk about aminosugars. Mostly I'm going to focus on synthesis routes that we developed in our own laboratory, which are cascade-based routes. And then in the last couple of slides, I'm going to mention some recent literature, not from our group, on aminosugars as antivirals and the possible relevance for COVID. So my group are involved in the design and synthesis of bioactive molecules. Mostly we work with carbohydrates. These are just some molecules that we have made in the lab. For example, we've been interested in immunostimulatory glycolipids, shown in the top left. We have been involved with Rob Woods at the CCRC in Georgia on the design and synthesis of influenza inhibitors, hemagglutinin inhibitors, conformationally constrained glycomimetics, like the one shown. We have been involved in basically new types of materials for probing carbohydrate protein interactions, model systems. So we have a manuscript which is currently in review, work done on a sulfatide model system, which has been done in collaboration with Hans-Joachim Gabius in Munich, as well as Virgil Perchich and Michael Klein in Philadelphia, where Janus glycodendromerosomes have been synthesized. We've also been involved in glycocluster synthesis, and the tetravalent compound shown presenting GalNAc residues is an nanomolar arranging ligand for a lectin called human macrophage galactose C-type lectin. And this is a lectin which is found on the surface of macrophages. It's involved in recognition of tumor antigens, and also it's involved in virus uptake. So these are just some of the projects. So we have been interested in the synthesis of aminosugars, and aminosugars are glycomimetics, and Roland Peters has already introduced a little bit about some of them. If we take a look at the structure of beta-D-glucopyranose, if we replace the ring oxygen with a nitrogen, we get an aminosugar. So shown on the top right there is nageromycin. There are different classes of aminosugars, such as the piperidine one shown with a 6-membered ring, but you can also have pyrrolidines or indolizidines and so on. They are naturally occurring compounds, and they have been, some of them have been approved as medicine. So for example, as Roland Peters mentioned, glycet was approved for type 2 diabetes. Also, there is Zavesca, shown in the bottom right, miglustat, which was an orally available drug administered for Gaucher disease. These are deoxy nageromycin. That means that we have no hydroxyl group at what might be the C1 position of the piperidine. These are more stable. And we can see in glycet and Zavesca, the nitrogen of the piperidine is alkylated. So in early work that was done in our laboratory, when I started out my research, we developed synthesis of deoxynageromycin and castanospermine. Castanospermine, shown in the bottom, is a indolizidine derivative. We use reductive cascade reactions. So for example, we basically synthesized 5-C-methoxyglycopyranosylazides as shown in the top part of the slide, and used reductive one-pot cascade reactions to generate the deoxynageromycin. So for example, when you take that kind of azide and subject it to catalytic hydrogenation, we presume that what's happening here is that the azide is reduced to the glycosylamine. This is a hemiaminal, so it can, in principle, open to a acyclic imine, which also now contains a hemiacetal, and the hemiacetal is not stable and will give you a ketone, and then reduction of the imine followed by cyclization of the reductive amination reaction, we can get deoxynageromycin. And in a variant of this reaction, we also synthesized castanospermine. So later on, we used other types of cascade reactions for the synthesis of aminosugars, and they involved the Huysgen cycloaddition reaction. So the azide alkyne click reaction is very famous reaction now, but you can also do an azide alkene variation of that. So when you get a cycloaddition between the azide and alkene, we can get a triazolene. So from D-gluconolactone, shown on the top left, we can synthesize this reactant, which has an azide and alkene present. And then the Huysgen cycloaddition gives you the triazolene. Now this is, unlike the triazole, is not very stable and decomposes to give you an aziridine, shown in the top right. And then the aziridine can react with, if there are nucleophiles present, to give you the protected piperidine derivatives. So in this way, if you use water or acetic acid as a nucleophile, we can generate, after removing protection groups, the deoxynageromycin or derivatives of deoxynageromycin. The reaction is stereoselective, and that is explained by, in what's shown in the bottom right-hand part of the slide. There are two possible transition states which can give rise to the piperidine, and the one on the left is more stable because we have minimized allylic strain, so that accounts for the stereoselectivity that's observed in this reaction. Now there are also interest in generating aminosugars with substituents at what might we call the anomeric position. So we can have a carbon substituent there. Aminoglycosides, shown in the top left, are unstable. So that's why a lot of work is done with the deoxy sugars, but we can have a carbon substituent. Their alpha homogermycin is a natural product, for example, and there has been quite a lot of interest in synthesis of these. So we were interested in the allylic azide rearrangement and using it in this context. So this is the allylic azide rearrangement shown here. It's a sigmatropic rearrangement. So we can have interconverting mixtures of allylic azides. The reaction was first reported by Gagnon, Winstein, and Young in 1960, and it has also been exploited more recently by Sharpless and Foken and some others since then in click chemistry. So we wanted to use allylic azide rearrangement in tandem with Huescan cycloaddition. So we could have a primary allylic azide, for example, which could be in an equilibrium with secondary allylic azides. We could have two stereoisomers, and perhaps one would react in a selective way. And we could generate C-substituted aminosugars with two stereocenters generated. And this was work that has been done by Lorna Moynihan and Rekha Chadha during their PhD thesis. So typical synthesis of precursors. So top left structure, 6-iodo mannose derivative is easy to prepare. So for example, you can introduce the acetonide at the cis diol, then introduce a Tes group at the C4 hydroxyl group. Then we have this iodo, and then you can do a zinc mediated reductive fragmentation reaction. This was work initially reported by Andreas Vasella and some work later done by Robert Madsen. This fragmentation reaction gives rise to an aldehyde, and when you do the Wittig reaction, we can generate the structure shown in the bottom left. And then you can reduce the carboxyl group to the primary alcohol and exchange that for an azide using a Mitsunobu type reaction with diphenyl phosphoryl azide. This gives us the allylic azide. Predominantly, it exists as the primary allylic azide with the trans stereochemistry and the double bond. And the rearrangement in conjunction with the Huescan cycloaddition can be carried out. So for example, on the top, we get the rearrangement followed by cycloaddition to give a triazolene, which can be observed or isolated. And then we break down the triazolene in the presence of acetic acid. We can generate in 35% yield the piperidine shown in the top right. And then removing the protecting groups and reducing the double bond, for example, we can generate this mannose derivative that's shown. If we use an acetonide on the bottom left, different acetonide, we can get a slightly better yield, but we get the same product ultimately. You can use thiophenol as a nucleophile. The structure shown in the bottom right is generated, and we can get an X-ray structure, and that can help us to confirm the stereochemical arrangement. And when we're dealing here with mannose derivatives, and it's not easy to distinguish between the alpha and beta anomers, and actually the beta compound is a natural product, but it had been incorrectly assigned. The structure had been incorrectly assigned in the literature, and we corrected that. Now, we also were interested to look at, could we make pyrrolidines? So we made precursors to give rise to pyrrolidines. The structures are shown here, which we synthesized. And we looked at the rearrangement followed by the cycloaddition. In this case, actually, when we use acetic acid as a nucleophile from this particular compound, we obtained actually piperidine, not a pyrrolidine. So the question is, but actually the reaction works very well, a 77% yield. And what's also interesting is we get an all-syn arrangement of the hydroxyl groups in the product. So just the pathway to explain the piperidine formation. So actually Rekha monitored this reaction in DMF. So in deuterated DMF by NMR. So this was, we could see clearly the formation of the triazolene. I show you HSQC spectrum where we assigned all the signals to the triazolene structure. If you continue to heat the mixture, we can then see the formation of the aziridine, and the signals in the bottom are indicative of that. And then we can add the nucleophile, and it's possible that the aziridine produced could react by kind of like SN1 or SN2 type mechanism. So really the outcome depends on the strength of the nucleophile. A stronger nucleophile will tend to form pyrrolidine. In the case of the acetic acid, what we got was basically the probably an SN1 reaction where we may generate a carbocation, which gives rise to an intermediate like the one shown. And we can see the diaxial arrangement. We have an oxygen substituent in the ring in an axial arrangement, and that may be because it helps to stabilize the cation. And then the nucleophile attacks from this side shown. And the reason we believe that happens is that maybe it gives rise to a chair-like transition state. And that explains the all-syn arrangement that's produced. With regard to what the efficiency of the reaction, the aziridine formation, the efficiency of aziridine formation is very important, and there's a competing reaction. So you can form an imine, can form competitively with aziridine, and if the imine formation is competitive, then very often we find the yields of the reaction aren't low, and we don't fully understand how we might alter that yet to get more efficient aziridine formation. So some of this work was featured on Synthesis a few years ago that we produced. And more recently, we've produced aminosugars with quaternary centers. So for example, the structure in the top left, we have an extra substituent on the alkene. So we make this from the mannose, and then the rearrangement takes place. Now we get a tertiary allylic azide, and then we get cycloaddition to form the triazolene, and then the aziridine, and followed by reaction with acetic acid, we can generate in about 60% yield the amino sugar with the quaternary center next to nitrogen. Now the methyl group, you can reduce this or take off the protecting groups and reduce, and we can see here is that the methyl group prefers to be equatorial. So the reason we believe the methyl group goes equatorial has to do with 1,3-diaxial interactions. So for students, you can think about the steric effects of substituents and cyclohexane. So the A value for a methyl group is bigger than the A value for a vinyl group, and so in the transition state, the methyl group will prefer to be equatorial rather than axial to minimize 1,3-diaxial interactions. So this work was published last month and featured on the European Journal of Organic Chemistry. So that's some of our synthesis work. Basically, I just want to mention in a couple of slides about D&J derivatives as antiviral agents, and how they work as antiviral agents is because they block endoplasmic reticulum glucosidases, so they are glucosidase inhibitors. So in virus replication, viruses use the glycoprotein synthesis machinery of the host cell to produce their own proteins, or their own glycoproteins. So this, for example, is how the famous spike protein of COVID, for example, is produced. So the endoplasmic reticulum glucosidases 1 and glucosidase 2, they play key roles in the biosynthesis and in endoplasmic reticulum quality control. So these glucosidases are involved in the processing of glycans, as well as in the folding and assembly of both host and viral glycoproteins. So if the glycosidases are inhibited by D&J derivatives, then the proper synthesis and folding of viral glycoproteins is blocked. And so that means that infected cells produce fewer infectious virions, and there's actually also a barrier to escape of viral mutants. So the aminosugars have potential in this regard, and I show both again miglostat and also a compound called UV4B, which has been under investigation. And in a recent paper in the Journal of Medicinal Chemistry, Nico Zitzman and collaborators at the Oxford Glycobiology Institute have studied UV4B. So it's a methoxy nonyldeoxynigeromycin derivative here. It's a glucosidase inhibitor. They have studied it not in the context of COVID, but in the context of influenza and in dengue viral infections. And what they found is that a single dose of UV4B prevents death in mice infected with lethal doses of influenza and the dengue virus. So compared with miglustat, I think you need multiple doses of miglustat are needed, whereas a single dose of the UV4B, but that it correlates with the inhibition of the endoplasmic reticulum, glucosidase 1, and they can start treatments as late as 48-hour post-infection. So this is an issue with the use of the glycosidase inhibitors as antiviral agents, is potentially, it may have to be used as prophylactics, but that can be still very helpful for people who are potentially exposed to infection. So this was work produced by Zitzman and co-workers, and then more recently there has been 2 papers arguing for the use of miglustat or as a repurposed antiviral agent. So one from Spencer Williams and Ethan Goddard-Borger, which is published in the Biochemical Society Transactions. I give links to it here, and there's also another paper by Alessandro Marcello and co-workers, which is a preprint. It hasn't been peer-reviewed as yet, but both argue for the use of miglustat. Reason is the miglustat or Zavesca is orally bioavailable. It's well tolerated. It's been used now for quite a while in the clinic. It's actually now available from generic manufacturers, and in the work by Marcello, they have proposed that it inhibits the folding and release of the COVID spike protein. And Spencer Williams makes a very detailed argument about the use of miglustat. So because it's available from generic manufacturers, it could be available for investigator-led trials, for example. So there's 2 possible candidates based on aminosugars as antivirals. So in summary, these are some of the structures that we've made over the years. I haven't talked about all of these today, but I've given the references or citations to the papers if you want to read more about that. We have D&J derivative that might be quite interesting for target macrophages. If anybody is aware of somebody who may be interested in collaborating in this context, we'd be happy to talk to them. And finally, I'd like to thank my funding, those who have funded me, particularly Science Foundation Ireland, the European Union, the Irish Research Council, and also we've had some industrial support from Pfizer and Roche and Lilly and Astellas over the years, and Pfizer are currently supporting us with a PhD student in the area of flow chemistry and aminosugars, so we're grateful to them for that. And I'd like to thank all the researchers in my group. This is some recent pictures of our group members. We are located in Europe, on the western fringes of Europe, west to the left of the United Kingdom and Ireland. So if you take a look in the top left there, Galway is located directly west of Dublin, and we're facing the Atlantic Ocean. So I'd like to say thank you again, and I'm happy to answer any questions.
Thank you, Professor Paul Murphy. It was an interesting lecture on synthesis of aminosugars and potential as antiviral agent.